Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It's my honor and privilege today to welcome our speaker, Dr. Chongzhi Zang, from the Center of Public Health Genomics at the University of Virginia. Chongzhi earned his BS in physics from Peking University in 2005 and his PhD in physics from the George Washington University in 2010. He undertook a postdoctoral fellowship in biostatistics and computational biology at the Dana-Farber Cancer Institute at Harvard University. Dr. Zang is an assistant professor in the Center for Public Health Genomics at the University of Virginia School of Medicine, and he holds faculty appointments in the departments of Public Health Sciences, Biochemistry, Molecular Genetics, and Biomedical Engineering at UVA. And he is also a member of the UVA Cancer Center and the UVA School of Data Science. Chongzhi is a computational biologist with expertise in cancer epigenomics. His research focuses on algorithm development for high-throughput genomic data analytics and integrative modeling of gene regulatory networks in mammalian cell systems. He and his laboratory are developing computational and statistical methods for analyzing high-throughput data from innovative omics technologies and using integrative data science approaches to study gene regulation in mammalian cell systems. He developed several popular computational tools for ChIP-seq data analysis, transcriptional regulatory inference, and multi-omics data integration. Dr. Zhang's lecture today is entitled Integrative Data Science Approaches for Studying Transcriptional Regulation in the Genome. And as always, we are streaming this lecture live and for recording via YouTube. If you are watching on YouTube, thank you so much for joining us. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Zang via the chat feature in their Zoom sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Chongzhi. Thank you so much for doing this. We really look forward to your lecture.
All right. Thank you so much, Jack, for the very nice introduction. So at the beginning, I wanted to share with you that I am actually an alumni from the 2018 cohort of the Biomedical Data Science Innovation Lab. This is our good picture taken from a beautiful resort in Bend, Oregon. It was an extremely fun experience. I benefit a lot from it and also receive the most gorgeous portrait of myself drawn by a fellow participant. So I really look forward to seeing each and every one of you, this year's participant in Charlottesville in a couple of months. So today I'm going to share with you about my research on computational approaches and data science approaches for studying transcriptional regulation genome-wide. So first of all, I would like to echo Dr. Don Brown's lecture from earlier in this seminar series about what is data science. Of course, data science is an interdisciplinary field of research. It lies in the intersection between the three areas. So I changed this picture a little bit, specifically to biomedical data science. So the three areas that form the foundation for biomedical data science includes math and statistics, which is the quantitative science basis, and computer science and information technology is the tool and engineering power for data science. And of course, we have the important domain knowledge and as the subject of research and the ultimate goal of doing all of the data science research. And in this particular context, is the biology and biomedical sciences. So data scientists require training and skills in all the three areas nowadays. Although most people who are doing data science research are usually from one of the three backgrounds, we usually need some knowledge and some understanding of all three areas, especially biomedical sciences, as this is where the ultimate problem and the solution are located. So another way of understanding data science is to start from data and using computational or other quantitative approaches to convert the data into information and connect them into knowledge and to look for insights and eventually look for new biology or new science from this data and information. So every step in this process involves new methods, and eventually our goal is to make scientific discoveries from this data. So data science is the combination of data and innovative computational modeling. And meanwhile, we also need to pay attention to the data problems, since they easily have overfitting and overinterpretation issues that we want to pay attention in the data science research. So I wanted to share with you a blog piece from my postdoc mentor, Shirley Liu. She wrote this in a few years ago about levels of bioinformatics research. I think it also applies to the general biomedical data science research very well. So that there are different levels of the research, and just like if you have watched Kung Fu Panda a few years ago, that would start from the level 0. Level 0 is not the type of work that we actually recommend, so-called modeling for modeling's sake. As this is as if you only have a hammer and you are looking for nails everywhere, and you need to get lost from this approaches, since you are not focusing on this particular biomedical problem. And the level 1 is to apply existing software tools to analyze new data and to make new novel findings. So this is the start level of bioinformatics research. They focus on the particular biology problem, and it's important to pick the right tool to do the job correctly. And this is also a good training at the start point of biomedical data science research. And for level 2 is to develop new methods for general data science problem, focusing on the research area. The important thing is that the tools and the methods or models should be useful to the field, to the study of a particular problem, or to analyze one type of data from a new technology. And they should advance the knowledge in this area, and they should be able to be used by other people in this field. And level 3 is to integrate in existing big data in a smart way to make new biological discoveries. So this is really requires a higher level of training and skills, because the data is there, and the data are usually generated by others, and usually the data are from multiple different sources. And the key is to use a right and smart way to integrate the data and to make new findings that cannot be seen by only looking at the one data set. And of course, the ultimate goal is still to make new biological findings. And lastly is the level X, is the senior level. To make a bigger impact in the field is to provide key integration and modeling to massive data usually from big consortium or team science efforts. And this type of work usually includes the integration of all levels from 1 to 3. I believe that a successful data science researcher or biomedical data science researcher is probably doing all levels 1 to 3 type of work, with the option and the opportunities to do some level X work. So after a brief introduction, I want to share you 2 stories of the research work that my lab is recently doing. One story is I categorize as a level 2 work, is to develop computational methods for transcriptional regulator analysis. And the other story is so-called level 3 work, is we use the publicly available data to study transcription factor CTCF, its pattern in the cancer genome. So recently we celebrated the 20 years anniversary of the publication of the draft human genome sequence. The completion of the human genome project did not mean that the effort of cracking the genetic code was done. On the contrary, it only marked the start of the modern genomics research. One of the most significant technological breakthroughs in the entire biomedical field in the past 20 years, in my opinion, is the development of high-throughput DNA sequencing techniques. Sequencing cost has dropped by over 1 million-fold since the Human Genome Project. DNA sequencing technologies was not only used for sequencing the genome, the human genome itself, but it has become a positioning or a profiling tool to study many different elements and factors associated with the genome. And the genomics has been involved to the question, solving the question of tackle how the genome works dynamically in different conditions and how this genome can create such a diverse cellular phenotypes. In eukaryotic cell nucleus, the genomic DNA is sophisticatedly packaged into the structure of chromatin. A number of protein factors, including histones with modifications, and transcription factor proteins that are interacting with DNA and other chromatin remodelers, they all have the function of regulate the gene transcription or gene expression. And the gene expression pattern in a specific cell defines the cell identity. So the high-throughput sequencing technologies can help measuring the genomic patterns of all of this information in the genome. For example, the transcription level can be directly measured by RNA-seq by measuring the RNA transcript generated through the process of transcription. And the protein factors, including transcription factors and histone modifications, can be measured by ChIP-seq that usually marks the regulatory regions across the genome. And if we don't know what particular factors to chip in the cell systems, we can measure the entire open chromatin by DNA-seq or ATAC-seq. And those enriched regions from the data covers most regulatory elements, regardless of what specific protein factor interacts there. And for both ChIP-seq and DNase ATAC-seq technologies, they mark the regulatory elements not only in the promoter regions that are nearby from a gene locus, but there are a lot of more are enhancer-like elements that are in the distance from the gene itself. It's worth noting that the most regulation events in human or mouse systems are at these distal enhancer elements. And the protein coding genes sequence only covers about 2% of the human genome. And 98% of the genome are non-coding, including those elements for regulation for gene expression and the chromatin structure. So the existing identified regulatory elements might cover about 15 to 20% of the genome, if not more. So as I just mentioned, the ChIP-seq and DNase and ATAC-seq, they can measure the genome-wide regulatory profiles of some factors across the genome. And for ChIP-seq, it's measuring DNA associating factors. And for DNase and ATAC-seq, they measure the open chromatin profiles. And they all use different biochemical assays plus the high-throughput sequencing technologies. So using the advanced emerging technology, we can generate a large amount of data from this that capture the information from different elements in the genome. That eventually we got the data and to visualize the data in a certain way across the genome. And an important important role of computational analysis or data science research is to decode this data, is to convert this data back and to retrieve the actual biological meaningful information, and even from the data to find a new biology that is currently unknown. So my lab develops computational methods and use this data science approaches to study transcription. So there are two main types of projects that are going on in my lab. We classified it as a level 2 and level 3 works, are method development and the data-driven discovery to identify new biology. For method development, for this part, methods, there are different levels of methods, in my opinion. So there are model type of methods. And in this context, the model means statistical models or mathematical or even physics models, including machine learning models. So these are the basic quantitative models that can tells the connections and the relations of different elements from the data. And on top of the model levels, there are computational algorithms that we can implement these models to make it happen. And then on top of the algorithm level, we also develop software tools so that the entire community and other researchers can use these tools and the models or algorithms we developed for their own research. So we will first share with you the first story, a method development story about a computational methods. We name it as BART, and you will know what it stands for in a minute, for transcriptional regulatory analysis. So as we know, gene expression determines the cell identity, and the regulation of gene expression is important topics in molecular genomics and genetics research. So gene expression profile or transcriptome profile can be measured by high-throughput technologies like microarray and more recently the RNA-seq and even single-cell RNA-seq experiments. A common practice of doing RNA-seq or gene expression analysis is first to identify the differentially regulated genes from a experimental system. We compare the control and the treated or different conditions, and these groups of upregulated genes or downregulated genes are sharing the differential expression pattern. And in other cases, we can use unsupervised clustering approaches to identify clusters of genes that are co-expressed among different conditions. This can be a large group of samples or even different clusters from a single cell analysis, single cell data. That means these each different cluster, each different groups or conditions might refer to different cell types, and each cluster of genes may represent the genes that are specifically expressed or active or regulated in a particular group of cells. So after identified these gene sets that share the expression pattern, a common question we want to address is that what transcription factors regulate such a gene set with similar gene expression patterns? There are some common types of approaches to address this issue, and all of them, they will all have some limitations for these existing approaches. So first and foremost is the ontology, gene ontology-based methods. There are a lot of databases like gene set enrichment analysis or gene ontology that you can characterize the function of a group of genes. But this is of course limited by the existing database. And the second type of method is co-expression based method, in which we use the clustering and the network type of analysis to infer the regulatory directions in the regulatory networks. That usually only depends on the gene expression data, but the expression of a regulatory factor does not always means the regulatory activity of that factor. And the third types of method is DNA sequence motif-based method, in which what we usually do is to look for the regulatory regions, usually in the gene promoter regions, and to scan for the highly represented sequence patterns, what we call DNA sequence motifs. And these sequence motifs may give us some information of what specific DNA sequence specific transcription factors are bound at these places to regulate the gene. But the occurrence of a DNA sequence motif does not necessarily mean that the transcription factor binds there. And in the human, mouse, and high order organisms, most of transcription factors binds at distal enhancer regions, which are difficult to identify from the beginning. And also, it's difficult to associate these distal elements to their target genes, as it's possible that multiple enhancers can regulate in the same gene in the same or different context, and one enhancer can also regulate multiple target genes. So the rationale of our approach, the approach we develop, is to make use of existing ChIP-seq data, which provides the direct binding information of transcription factors in a lot of different cell conditions and systems. So since the development of ChIP-seq technology in 2007, there are increasing amount of ChIP-seq data that are becoming available in the public domain, and numbers are also growing a lot. And I want to mention here that we probably heard that many of the publicly available data are generated from the large consortium like the ENCODE project or Roadmap Epigenome Consortium. But in fact, the data that are generated from this consortia are only consist of less than 10 to 20% of all the data that are available in the public domain. There are thousands of labs are generating their own individual data that are also available. So this is actually a much richer data resource than we thought. So and this plot also shows the increase of different types of histone modification, histone marks ChIP-seq data available. And in this particular project, we take advantage of several types of sequencing data. That includes, we want to use the identify where are the regulatory regions or possible enhancers. In this case, we use the DNA-seq data, since this is from a few years ago, and there are over 500 data sets from different DNA-seq from different cell types. As in the recent years, ATAC-seq has become a more popular approach for to profile accessible chromatin, and ATAC-seq data may have even more data shortly, or probably already. There are more than DNA-seq. And a second type of data we use is to quantify the enhancer activity, is the ChIP-seq data for one type of histone modification, H3 lysine 27 acetylation, K27 acetylation data. And because this histone modification marks the activity of active enhancers from other histone marks. And from this plot, you can see the amount of H3K27 acetylation data are increasing faster than some other histone marks. It's probably because people becomes more interested in this factor that regulates the enhancer regulatory activity. And we also have collected over tens of thousands of ChIP-seq data for over 900 different transcription factors. And these are the data we want to use. So our strategy, this method, is to, there are two-step strategy in this method. We named BART, which stands for Binding Analysis for Regulation of Transcription. So the first step in this strategy is we give a gene set as input. We first wanted to use the DNA-seq data and the H3K27 ChIP-seq data to infer a genome-wide cis-regulatory profile that mark the active enhancers that could regulate the gene sets. And the second step in this strategy is to integrate this inferred cis-regulatory profile with the over 13,000 data sets for transcription factors to identify what transcription factors are binding at these enhancer regions that eventually regulate the gene sets. So the first step of the regulatory profile inference, in the first version, we take advantage of an earlier model that we developed named March. We always love to give fun names to the tools and models we develop. And the idea is to convert the hundreds of or over 1,000 H3K27 ChIP-seq data from different cell types and different conditions, and to give a quantitative measure of the regulatory potentials on each gene, and then use a regression approach to do a feature selection and to select the informative samples that can capture the regulatory profiles that are specific to the input gene sets we are interested in, as compared to the other genes in the genome that we are not interested in. So in fact, later on, we updated this method with a different regression approach using adaptive lasso. And this new updates is actually, the idea come from our discussion at the Biomedical Data Science Innovation Lab a few years ago with a fellow participant in the Innovation Lab, Professor Yang Chen, who is a statistician from University of Michigan. She suggested that adaptive lasso might be a better approach of addressing this. And indeed, we updated this method in our recent new version of BART. And the second step is to associate the inferred cis-regulatory profile with the tens of thousands of ChIP-seq data, and to identify which data sets and which transcription factor that have the binding profile that are most associated or most similar to the cis-regulatory profile. And we use several additional steps of integration and remove some background, and to get an add additional level of statistical test to give a integrative rank of all the transcription factors that have ChIP-seq data. And eventually, we make the prediction by saying that the highly ranked transcription factors from the entire list are predicted or inferred to be the most likely transcription factors that regulate the target gene sets. And we compare some of the existing methods for the transcription factor inference. Some of them are using the motif-based approach, including Homer, and some others are also use similar ChIP-seq database approaches to evaluate the performance of these different methods. The evaluation is done in data from a database called NOCTF, in which they contains the gene expression, differential expression data from a knockdown or a knockout of some transcription factors. And we say if we don't know what the transcription factor it is, if we only fit in their differentially expressed genes, whether we can correctly predict the true regulator that was knocked down to generate that data sets. And in they include, the database includes over 500 data sets for about over 300 different transcription factors. And for both the data sets, number of data set level, and the number of factors level, our new version of BART, in the web version called BART Web, outperforms several other existing methods, also outperform our own earlier version of BART in identifying the true factor from the knockdown data sets. Then we also thought about if this tool can infer the transcription factor that are associated with a regulatory profile, it may be also be able to apply it to other type of genomic data if we also want to be interested in the transcription factor association. So recently we developed a variant version of this of BART. We named the BART 3D. 3D means the 3D genome data. And the goal is to identify the transcription regulators or transcription factors that are associated with differential chromatin interaction from a differential high-C data analysis. So high-C measures the chromatin interaction across the genome that can help us build up the entire three-dimensional structure of the genome and chromatin structure. By comparing two Hi-C maps, the goal here is to first identify the genomic regions that have a different level of interaction with their nearby regions within a certain distance boundary. And then to infer the transcription regulators that are associated with this differential chromatin interaction profile. So then we test this method, and it looks like it works well on identifying the right transcription factors that are knocked down from either the knockdown or deletion of transcription factors, knockdown of the transcription factors and the Hi-C data generated afterwards. And we found that the BART3D can not only identify the factors that were knocked down or knocked out, but they also identify some other interesting factors that are either can be proved or need to further validation to be also play a role in this process of changing the chromatin conformation structure. So as a result, we integrate all of these several tools we developed into a large system, including a web server that can provide the analysis by any user. Even for a non-computational biologist, once you get your gene set, you can simply do the analysis from this web server yourself. So this BART tool can accept multiple type of inputs, include a gene list that was designed in the beginning, and it also be a ChIP-seq profile if we want to identify what other cofactors or other factors that are close to this ChIP-seq profile, or even a tag-seq profile. And it can also accept any region from the genome, and they can tell you what factors binds there more likely than another method. And Hi-C maps use the BART3D approach. So we hope that this tool can provide an alternative or complementary approaches to other existing methods, like including DNA motif scan. So hope that these tools can help your studies of the functional regulators to explain or interpret your differential gene expression data or other type of genomic profiling data. So then we want to look for the find some applications of the BART tool ourselves. So one effort that we did recently is to predict the active transcription factors in cancer type using the existing cancer transcriptome profiling data from the Cancer Genome Atlas, or TCGA. Because of the limitation of the availability of primary tumor samples, TCGA does not provide any ChIP-seq data that measure the actual regulatory profile from this cancer patient. But TCGA has RNA-seq data measuring gene expression. So for each and every cancer type, we simply did a differential expression analysis to identify the cancer, comparing the cancer samples with the normal samples to identify the differentially expressed genes, up-regulated genes in cancer and down-regulated genes in cancer, and then run the BART analysis to integrate the information that we have from the BART, from the large amount of data from the public domain, to use the public data to infer the possible transcription regulators, transcription factors that plays a role in the cancer gene regulation. And this is actually done by a very talented undergraduate student in my lab, Zach Thomas. And he processed all of the data from TCGA and displayed this data to the public domain in a database as a resource. And in this way, we can browse or look for the activity of each and every transcription factors that have ChIP-seq data. So for example, if we look at the pattern, the TF activity pattern of one transcription factor, in this plot, we are plotting this BART prediction score for this factor for in each and every cancer type. And we also compare to a earlier another similar approach that using a different similar work, but using a different approach to infer the transcription factor from cancer. That's a work from Systrom Cancer database. And by integrating these two elements, also including the expression level of the transcription factor itself, we can see that if this this is a big circle in the top right, which means that they have a high score in both measurement as well as a high expression level of this factor in colorectal cancer type, which means that this transcription factor may play a role in this cancer type. So this can be useful in the study of each and every cancer type, and useful for study the activities for each transcription factor. And we are very happy to share with you that after the publication of this method, there are other labs who are using our tool, BART tool, to find the important transcription factors in different cancer systems, and they have very good publications, including in Nature, and another work in this is from another lab at UVA that study glioblastoma, and this work actually got semi-finalist in the recent Stat Madness competition, and we are very excited that the computational tools that we develop can help other biologists in their studies. So as a quick summary for story one, we developed this tool called BART. It's a regulatory factor, transcription regulator identification method. And we leverage a large collection of public available ChIP-seq data to build the model, and they can be applied for analyzing multiple types of data. And eventually we developed open source software tools and web servers and other database resources with application to cancer research. Hope that these tools can be helpful for any researcher who are interested in the transcription regulation as well. And next, I will use a few minutes to share my second story, a level 3 data-driven discovery story about CTCF pattern in the cancer genome. As I mentioned briefly, that the genomic DNA is organized in a hierarchical chromatin structure in the eukaryotic cell nucleus, and the three-dimensional hierarchical structure includes the multi-megabases sized compartment, AB compartment, and hundreds to megabases, 100 KB or to megabases size topological associating domains or tags, and numerous smaller chromatin loops. So CTCF is a protein, DNA binding protein, or also categorized as a transcription factor that binds in the DNA, and they can induce DNA looping with cohesin complex and other proteins, such as mediators. It binds at most task domain boundary regions and has an important function in maintaining the chromatin structure and the higher order topology. So CTCF as a transcription factor is the very factor that connects the local level transcription regulation and the larger scale chromatin structure. So that's why we're specifically interested in the pattern and behavior of this transcription factor. And we found that CTCF binding is usually very stable and conserved across different cell types in the human genome. This is expected because it binds to maintain the relatively stable chromatin structure. So we compare the chromatin accessibility profile from dozens of cell types from the ENCODE consortium, and we found that the variation of chromatin openness at CTCF binding sites is actually the smallest as compared to the other non-promoter or enhancer regions and the promoter regions. So for such a stably bind protein, a disruption of CTCF binding sites can be very dangerous to the cell. It can change the chromatin conformation and interactions that alter the gene expression pattern. So existing works usually attribute the CTCF binding alterations to DNA sequence mutations and the DNA methylation changes, as many individual cases have been found in the cancer and developmental diseases. However, we wanted to take a look at the bigger picture view of the CTCF binding landscape and look for general pattern of CTCF binding alteration in the whole genome and in different cell types. CTCF binding can be measured by ChIP-seq across all different cell types. We collected over 800 public available ChIP-seq data from hundreds of human cell types, including both normal tissues and disease, including cancer cells. From this snapshot, we can see that across different cell types, some binding sites are very conserved, and with some other binding sites are specific to several cell types. So we kept over 700 data sets that have high quality and merge all the identified binding sites together and generated a whole repertoire of possible CTCF binding sites in the genome that includes that have over 600,000 of unique regions in the genome that can have CTCF binding in any cell type. And then we look at for each binding site, we look at the occupancy score, meaning that how many of the data sets where there is a peak at this region. And then we look at the distribution of these occupancy scores for all the CTCF binding sites. We can see that the majority of the binding sites are specific to a few data sets. They are cell type specific binding sites. Where on the other side of this curve, we observe an elevated tail. There is a group of binding sites that stably occur in almost all data sets. Imagine a background where CTCF binds randomly at different sites in different cell types. The expectation of observing a peak binding sites with certain occupancy score should decrease drastically when the occupancy score increases. Then we used a power law function to model this background, and we can see the number of conserved sites are much greater than expected by chance. So we define this group of binding sites are constitutive binding sites, and they are the group of binding that are marked at topological associating domain boundary regions. Once we have all the CTCF binding in the human genome, we want to know if there are any binding sites that are specific to one cell type. And in this context, we are interested in a cancer. And we use one cancer type, T-cell acute lymphoblastic leukemia, as an example to show our approach. In our collection, we have 8 ChIP-seq data sets from the TLL cell lines, and we compare the occupancy score of all the over 600,000 binding sites in the genome, and plot this distribution according to their occupancy score in the 8 TLL data sets, whereas the occupancy frequency in all the other 763 data sets. So for each element, the dance represents the number of ChIP-seq binding sites that belongs to this group. So we are specifically interested in 2 groups of CTCF binding events. One group is that the constitutive binding site, which means that they are conserved in most cell types, they bind there, but specifically lost in TLL, has a very low occupancy score in TLL. And whereas we define this group as a putative loss site. And on the other side, we define another group as putative gain site, which means that they are very cell type specific. They don't bind in most cell types, but very conservatively bind in the T-cell leukemia data sets. And we also make sure that these, for cancer studies, we also make sure that these gain and loss are indeed have different level comparing the cancer versus their matched normal cell type to make sure that they are indeed cancer specific, but not only cell type specific. Using the similar approach, we are able to identify such binding gain and binding loss in every cancer type that have enough available data, including leukemia, breast cancer, colorectal cancer, lung, and prostate cancer. So because most of the existing ChIP-seq data are generated from cell line models, we want to make sure that this binding sites actually occurred in the actual cancer primary tumor patient, cancer patient data. So not an artifact from the cell line. But as we know, we don't have ChIP-seq data for CTCF for most cancer patients, but we know that the chromatin accessibility are positively correlated with the CTCF binding. So we take advantage of the available ATAC-seq data from the TCGA to see if the CTCF binding loss or gain also potentially occurred in patients. So the idea is that we identified those gain and site, and we look at their differential chromatin accessibility levels comparing in the cancer and the other samples as control. Indeed, we found that these identified cancer-specific gain and loss events show a differential level of chromatin accessibility in the primary patient samples. That indicates that this unique loss or gain CTCL binding events are not only from cell line, but also occurred in primary cancer. So then we wanted to ask whether these specific binding events correlate with other features in the genome. So use our developed BART3D approaches that can measure the differential chromatin level. We found that the gain and the loss of CTCF binding positively associated with the gain and the loss of chromatin, while the elevated or decreased chromatin interaction levels, comparing the cancer and the normal cell type. And we also found that as previously studied, the loss of CTCF binding largely correlated with the increase of DNA methylation at the binding sites, also using the publicly available DNA methylation profiling data. But interestingly, we found that the DNA methylation, the gain of DNA methylation for loss of binding, but the loss of DNA methylation can only explain only a portion of the gained CTCF binding. They don't explain most of them. So now we wonder why are those binding sites specifically gained in this cancer type, whether there are other factors that could induce this event. So from the gained binding sites we identified, we first did a motif scan, but we did not find any other interesting motif other than the CTCF motif itself. And then we look further across the genome by using the high-C data, and we look at the regions that have an increased interaction with the gained CTCF sites. And then did use the BART, the tool, here comes the application of the BART tool to identify what possible transcription factors are enriched in these regions. And interestingly, we found several transcription factors that are known to be the oncogenic transcription factor in this specific cancer type. With the help of our experimental collaborators, we validate our prediction or to study what's the role of this transcription factor, namely Notch, in the occurrence of CTCF binding sites that are specific in this cancer type. So we perturb the Notch by creating a Notch off and Notch on states using a gamma secretase inhibitor, and to remove this inhibitor. And once the Notch binding is removed and regained, we wanted to see whether the identified gained CTCF also changed. And indeed, we found that after Notch is removed, most of the gained CTCF binding decreased. And after Notch is rebound, most of the decreased CTCF binding regained their binding in the cancer site. This means that the perturbation of this transcription factor can result in the change of the CTCF binding. On the other hand, we want to address whether the gain binding of CTCF is required for the transcription regulatory role of the Notch, the transcription factor in this cancer type. So we knock down CTCF using shRNA and generated the RNA-seq data to look at the expression. And if we use BART to identify the possible regulators for the differentially expressed genes after CTCF knockdown, the top enriched factor are actually Notch, which means that once you knock down CTCF, Notch target genes are down-regulated, as also shown using a previously identified Notch targeted gene sets. This means that indeed, CTCF binding is required for the oncogenic Notch to activate their Notch target genes. So although we did not observe a direct protein-protein interaction between CTCF and the Notch, this process is probably not a conventional protein recruitment from one to another. So we further proposed a model. This is largely still speculative and as a hypothesis that can be tested further. That we found that our idea is that the function of CTCF in this particular cancer cases is to facilitate the formation of a transcriptional condensate with the super enhancer regions for transcriptional activation. So Notch can be recruited by this large condensed transcriptional condensate induced by some transcription factors, including Notch that is known to induce super enhancers, with other cofactors and chromatin remodelers to create this very active transcriptional activation status to make the transcription happen. So in summary for the second story, we use the integration approach and take advantage of over 800 public available ChIP-seq data and also integrated with other genomic data. Most of them are available from the public domain. And we found the interesting pattern of CTCF binding in the genome of cancer cells. And we found that DNA mutations or DNA methylations cannot explain most of the cancer-specific gained or lost CTCF binding events we observed in cancer. And possibly the cancer-specific gained CTCF binding has a role of facilitating the oncogenic transcription activation by contributing to the formation of transcriptional condensates with super enhancers. So by sharing with you these two stories, I hope I've convinced you that our approach of doing data science in genomic research and using innovative data science approaches, we can change the omics studies that were used to be criticized by Sidney Brenner a long time ago as low input, high throughput, no output, but change this to a real precision science for precision medicine research. So last, I would like to thank the people who actually did the work, my lab members, and especially my postdoc, Zhenjia Wang, and my former student, Wenjing Ma, and the current undergrad students, Zach Thomas, who did most of the works for developing BART, BART 3D and the BART cancer resources. And Zhenjia Wang also did all of the work in the CTCF study. And also want to thank my collaborators, including Panos Christos from Northwestern, who helped us the experimental part for the CTCF project, and Yang Chen that I mentioned. We met at the Biomedical Data Science Innovation Lab, and she gave us important ideas of improving BART, as well as other collaborators and my funding agencies. And I want to mention that we received the support from the JKTG Foundation as a seed award as an outcome from the Data Science Innovation Lab that helps the development of this research. All right, So this picture was taken before the pandemic, and we really hope that we can get this pandemic over with pretty soon and can meet each and every with you in person very soon. With that, thank you very much for your attention. I'd like to answer your questions you have, any questions you may have.
Chongzhi, thank you so much. That is a fantastic amount of work. Really what you're doing there with these computationally intensive techniques for looking at transcriptome factors is really quite amazing. And it's fantastic that you have these resources that you've built, like BARTcancer.org, and places where people can actually take their own data or data that's publicly available and analyze it. I've just a couple of questions for you. Speaking of computationally intensive, how computationally intensive are these types of analyses, and how do you do it? Do you do it on local machinery and in your laboratory, or could one do it on local machinery, or do you have to use large scale high performance computing or cloud computing? What do you recommend?
Good question. So most of the work that we are doing is using high performance computing that are available to us from UVA. And some of the basic analysis of the data can be done using a local computer, but I would highly recommend using high performance computing for all the model building and model training, all of these type of work. They may be too intensive for a laptop. So this is actually the motivation for us to create such a web server for the general users to use the tools, because not everyone is has access to this high performance computing resources, and we want to make the tools as accessible as possible. So the web server we build up connects, it has a front end of a web server, but connects to the high performance computing in the background. So when the actual user submit the job to the web browser, web server, they actually, the HPC will do the computing for them and generate the results to the users in a fairly small amount of time. And I want to say that this is after a large amount of work of model training and the data cleaning up. So that may perhaps take more time. So we already compress the data into a form that only require the minimum amount of HPC resource.
Are there any special things that people would need to know in order to kind of get started using these tools, or is there any specific data science they need to know?
Yeah, well, as I briefly mentioned in the beginning, in order to understand how the tools work, one only needs to understand the basic statistics and some machine learning. That's all they need. But if we simply want to get the results from a data analyzing your data, our goal is to make it anyone can use. So even if you don't know any data science, you don't have any data science background, but you have the specific biology question, you have the data, and you can generate the result yourself. Of course, understanding the background will be helpful for data interpretation, but that's not specifically required for using the tool itself.
Chongzhi Zang from the Center for Public Health Genomics at the University of Virginia. Thank you so much, Chongzhi. Really appreciate you sharing your amazing amount of work with us and telling us about these tools. And fortunately for me, I don't have to understand any of it. I can just start going to use it. I hope that others find them also equally useful for their work. Thank you again, Chongzhi. Everyone have a wonderful weekend, and we will see you all next week. Thanks again. Bye-bye.
Thank you very much, Jack, and thanks everyone for your attention. Bye-bye.